Okay. Today what we're going to be talking about is meiosis. Let me grab my notes. So the first thing we're going to do is up at the top, we're going to define what meiosis is. I'm going to kind of drop down so I'm not right over this box.
Are we still on the same page?
You need to be writing on this page. Up at the top. Meiosis is going to be the process by which gametes, what do I mean by gametes? Does anybody know what those are? We talked about that. I can't remember. Gametes are sex cells. What are the sex cells?
Egg.
Right, egg and sperm. The process by which sex cells, gametes, are formed but what's special about meiosis is we're going to take the number of chromosomes and we're going to do what with them?
Divide.
Divide by
Two.
We're going to cut them in half, just like you read about diploid and haploid. So we're going to reduce them. We're going to reduce the number to half. So, for example, how many chromosomes do humans have?
46.
46. So our haploid number after meiosis is going to be 23. Now, the way this is going to work is on your paper, you've got kind of different blocks, and they're all numbered. There's a lot more steps to meiosis than there are to, what was that thing we just got done with? Mitosis. So what I'm going to do is I've got kind of a block up here, and you're just going to kind of follow along with me. So we're going to start up in the very top left-hand corner. This is going to be the first step. And it's going to be, meiosis happens in two major steps. There's meiosis 1 and meiosis 2. And so we're going to, today probably all we're going to get to is just doing meiosis 1 across the top. So just like in mitosis, let me try and make this match yours. So this will be the first step of meiosis 1. Just like in mitosis, we're going to start out with a cell that's in the normal, everyday part of the cycle, interphase. Very good. So I'm just going to draw a cell in interphase. And I'm going to label it interphase. Now, what is it that the cell needs to do with its DNA before it can start dividing? Replicate it. So we're going to go ahead and do that. We're going to replicate the DNA. So I'm going to write that the DNA replicates. What phase of the cell cycle is that?
S.
The S phase. And that's all normal, just like happens in mitosis. And what I'm going to do is I'm going to say, just to keep it simple, instead of having 46 chromosomes, I'm going to say that this organism has 4. Because it would be a lot easier to write 4 chromosomes than 46, right? So I'm going to start out by just writing that it has 4 chromosomes. Now, would that be its diploid number or its haploid? Diploid. Because the whole point of this is we're going to cut that number in half. So what's the haploid number going to be? 2. And I'm just going to draw, this is not really totally accurate, but I'm going to draw 4 chromosomes like this. And then when we replicate them, it's going to make what shape? What do we usually see the chromosomes as? A nice little X shape. So we start out with 4 chromosomes, and the reason I'm drawing it this way is I want to make a distinction for something. After replication, these are going to become 4 replicated. What do I mean by replicated? They are the same, but there's copies, right? Chromosomes. And that's where we're going to have the 2 sister chromatids together, correct? Correct. Thank you for making sure I was right. Now, each one of these chromosomes represents 2 sister what? And are they identical to each other or different? They're identical. They're exactly the same because they're copies. Now I'm just going to draw a line and I'm going to use the bottom half of this to do. Technically, that's not the first step of meiosis. That's just what we got to do to get there. We got to replicate the DNA. So the first step of mitosis is what? Prophase. The first step of meiosis 1 is prophase 1.
Wait,
So
wouldn't that only be 2 replicated chromosomes? Because there's only one on mine.
we got 4 chromosomes, right?
Uh-huh.
And now we have 4 chromosomes that all have 2 copies. There's 2 copies of each.
Oh, I get it.
You got it. All right. So we're going to draw. Let's draw. I'm going to kind of draw a big cell right here, but I'm not going to put anything inside of it just yet. And I'm going to label this as prophase 1. What are some things that happen in prophase and mitosis? All right, the chromosomes form. What else happens? Yeah, what's something else that happens?
The nucleus breaks down.
All right, nucleus breaks down, nuclear envelope goes away, the spindle starts to form. Those same things are going to happen in prophase 1, but there's going to be a little difference. So I'm going to go ahead and I'm going to draw my centrioles. And what's coming out of the centrioles?
Spindle fibers.
The spindle fibers. But when I draw my chromosomes, I'm going to draw them in a specific way. I'm going to use 2 different colors. You probably don't have 2 different colors, but that's okay. How many chromosomes do we have in this animal? 4. So I'm going to draw 2 chromosomes in blue. And right next to each of them, I'm going to draw 2 chromosomes in red. Why do you think I'm making that color distinction?
Daddy and mama said, you don't color
Right.
people.
Well, I'm showing that these blue ones are chromosomes. Let's say this is my cell, and it's going through meiosis. Those blue ones represent chromosomes that I got from my dad. And the red ones represent chromosomes that I got from who?
Your mom.
From my mom. But they're going to line up together. In other words, I'm going to have one from dad and one from mom that are going to match up with each other. And then, again, I'm going to have one from mom and one from dad. They're going to match up with each other. And we call these homologous chromosomes. What does homologous mean?
They match up.
What does homo mean? That's what you're asking. It means the same. So, in other words, these are chromosomes that have the same genes on them, but one of them I got from mom and the other one I got from dad. So if on this chromosome right here, if it has the gene for eye color, the other one does too. The only difference between them is that one of them came from one parent and the other came from the other parent. So in prophase 1, homologous chromosomes pair up, and we call that synapsis. What do we say homo means?
Same.
The same. So they're exactly the same. The only difference is, well, they're not. I guess I shouldn't say they're exactly the same. For example, let's talk about eye color real quick. Let's say that on these chromosomes, there's a gene for eye color. My dad may have one eye color and my mom may have a different eye color. And so I may get 2 different genes from my parents. But still, the gene for eye color is located on this homologous pair. Did I lose you? Yes.
You get 2 different eye colors.
I was thinking that.
There are some people that can happen to, but that's not normal. That's not anything to do with this. That's like a mutation. So in prophase 1, just like mitosis prophase, but when the chromosomes show up, they pair up in homologous pairs, and we call that synapsis. One way to remember it is they synap together.
Yeah, that's good.
I didn't come up with that, I promise. That was the lady in the back of the room. How many chromosomes we got per cell? How many chromosomes we got per cell? Right now?
4.
Okay. During prophase 1, there's a thing that happens called crossing over, and we're going to talk about that in more detail later. So right now, we're just going to write down that it happens here. Crossing over occurs. And I guess if we want to define it, we can say this is when there is a mixture of mom and dad's chromosomes. No, that's it for this box. That's it. That box is full. We're going to find out later that when these homologous chromosomes pair, they actually swap pieces with each other. So there'll be kind of a mixture of DNA that came from mom and dad on each chromosome after that's done. So this is all prophase what?
1.
Prophase 1, which happens in meiosis
1.
1, which tells us that there's probably going to be a prophase
2.
2 later.
Maybe even a.
Meiosis.
There won't be a prophase 3.
I didn't say that.
Okay, everybody got this? Can I erase this? Is there a way I can just erase this whole thing at once?
Circle it and touch it in the middle. But you have to do it right.
But I have to do it right?
You do it perfect.
Alright, here we go. I'm going to do it.
You messed up.
Did I mess up?
Oh yeah, you messed up.
No, I didn't. Okay, so that's the first step of meiosis 1. The second step is probably going to be called what? Prophase 1 is going to be
Metaphase 1.
Metaphase 1. So I'm going to go ahead and down here in the corner, you guys have a step 2 on that next box. And I'm going to draw a cell up top, like so. And then under it, I'm going to label that this is metaphase what?
1.
Right, metaphase
Why would
uno.
it say metaphase?
Because we have to distinguish between metaphase of meiosis and metaphase of
Mitosis.
distinguish. Look at that vocabulary word. What did we say this thing was here at the end?
Spindle.
The spindle and then the centriole, right?
Mm-hmm.
Now, what happens in metaphase of mitosis? What do the chromosomes do?
They line up in the middle.
They line up in the middle, and they're going to do that here, but they're going to line up as homologous pairs. As pairs. So instead of don't draw what I'm drawing. In mitosis, they would line up this way, straight up and down. Correct? That's not what happens here.
They just got a big
In meiosis, they line up in the middle as homologous pairs. You see the difference?
You got it?
So you only have 2 spindle
Right. Well, there's bunches of them everywhere, but there's only 2 that connect. Right. So this is the way we need to draw it. So I guess if we wanted to write on the side, they line up as homologous pairs. So this is a homologous pair. And what's significant about the homologous pairs is we got one of them from mom and the other one from dad. If we want to define this, we can say that the homologous pairs, I'm going to put this word in parentheses. Anybody know what tetra means? What that root means? What number it means? 3?
I thought it was that game.
7.
4.
Tetris is like
4.
game where you line up the shapes.
The reason, you don't have to draw this, but the reason they call it a tetrad, if I put a homologous pair up here, how many sister chromatids do I have total? 4, right? I have 1, 2, 3, 4. So sometimes you'll hear this called a tetrad. Homologous pairs line up at the equator. So if we want to write that in the box, we could say that this equals 1 tetrad. Or a homologous pair. Homologous pair and tetrad are just synonyms. They're just interchangeable. Guys, if you can just get this part right here, this is the hardest part to remember, I think, in meiosis, is that they don't line up on top of each other. They line up in pairs.
What's that little squiggly line beside tetrad?
That's a 1. I'm sorry.
Two.
I was so confused.
You're running out of time.
2 up. Oh no, we're running out of time. Well, I guess we'll have to.